Hey everybody, and welcome to Chapter 3. So in Chapter 3, we're going to be talking a lot about how the eye works with the brain. So you may remember Chapter 1, we talked a lot about, we basically laid the groundwork for a lot of sensation and perception as a class. We talked a lot about definitions and concepts, things like that. Chapter 2 was all about anatomy, right? We talked a lot about the eye, but didn't really talk about how the eye changes what we see. So we talked a lot about sensation, not all that much about perception. Chapter 3, we're going to be getting into perception, which means that we're going to be talking about the brain as well. So we're going to be looking at the very, very basics of vision. And what I mean by that isn't rudimentary colors or shapes. We're talking more basic than that. We're talking lines. We're talking about the difference between noticing light and no light. We're talking about edges and contours. We're going to work our way up to talking about shapes in Chapter 4 and then to color in Chapter 5. Alright, so in this particular chapter, we're going to be talking about a lot of things. I'm going to start out this video by talking about anatomy. So we're still talking about anatomy, but we're going to leave the eye and talk about the different parts of the brain that are going to help us perceive visual information. So this particular image that you see, I'm not going to say who it is, but chances are you know who it is. We're going to look at this as an illusion a little bit later in this chapter to help illustrate some of how we perceive lines and how we perceive basic gradients of black and white. So as we're talking about anatomy, this is a cool video that you can check out online. That talks a little bit about the eye and how the way the eye is shaped sets certain limitations for what we can perceive. And this particular video is referencing a movie in the Lord of the Rings series where this particular man, Orlando Bloom, is able to spot people in a field really far away, and it talks about whether or not he would actually be able to do that given his particular set of eyes and how big his eyes are. Alright, so let's get into anatomy. So where we left off last chapter, we talked a lot about the retina, right? That was where we ended up. We were talking about the retina, and we were talking about how it has these specific neurons that are going to be sensitive to light. Whenever they are exposed to light, they fire, or they fire more quickly, or they fire less quickly, or they stay the same. So we're going to continue from there. This is where we're picking up now. So here are our eyes, and these little pink strips right here, that is our retina. From there, our retina is going to feed backwards to the back of our brain. You may remember from Chapter 1 when we talked about the occipital lobe. The back of the brain is where we are going to really perceive where we are going to process a lot of that visual information. So here we have the left and right eyes. You can kind of see that these optic nerves, because remember we have these blind spots right here in the back of our retina, they are going to feed to basically a crossroads. This crossroad is called the optic chiasm. The optic chiasm is this big X in our brain where some visual information is going to feed over to the right side of our brain, some information is going to feed over to the left side of the brain. And that all depends on where that information was displayed in your retina. So what you're looking at right here is this light pink is the left visual field, and I have that incorrect. This is kind of confusing. This light pink is actually the right visual field. The dark pink is the left visual field. How do you know? Because if you're looking at this pink right here, what is this pink seeing? It's seeing, and I can't draw lines unfortunately, but it's seeing this information over here in this direction, which is the right side of our visual field. Now, it's on the left part of the eye, which makes it really kind of confusing, but you can kind of see, given the shape of the eye, this part of the eye is seeing things on this side of the screen over here. So that's just kind of how that that works. But basically, you can kind of see that stuff that is in the right visual field is going to end up staying over here in the left hemisphere. Stuff that is in the right visual field, this dark pink, is going to, once it gets to the optic chiasm, move over into the other. So this is the left visual field is going to end up in the right hemisphere. So in other words, it's contralateral. That is the phrase that we're going to use to talk about this and a couple of other things. Whenever you have something on one side of your body being handled by the other part of the other side of your brain. We talk about this a lot with movement. So your left hand is controlled by your right hemisphere and your right hand is controlled by your left hemisphere. It's called contralateral. We'll talk more about that later this semester. So once we get past the optic chiasm, this crossroad, this junction where we decide what information is going where, we're going to move over to the LGN. And the LGN is going to be another area that is going to help kind of filter and transport information to other sides of the brain. We'll talk about that in just a moment. So once things move over from the LGN, we're going to move straight to the back of the occipital lobe here in what is called the V1, also called the striate cortex. So this part right here, this purple part, this is where the first real visual processing takes place in the brain. This is where we are really starting to make sense of what it is that our eyes are detecting. So it's called the striate cortex. You'll also hear people call it the primary visual cortex. You'll also hear people call it the V1. V1 just meaning visual 1. So you're going to hear it called a lot of things. It's really important. It's going to come up a lot in this chapter and the next chapter as well. But that is here in the back of our brain. And then basically from there, it kind of blossoms out. That information is going to spread through the rest of the back of our brain as it moves to higher areas of functioning. So most of what we're going to be perceiving back here in our striate cortex is going to be boring stuff. It's just lines. It's just lines and curves and edges and things like that. It's not really until we get to some of these other higher order, more complex areas that we're going to start really perceiving shapes and things like that, which we'll talk about next chapter. So this flowchart gets kind of intimidating, but it does a pretty good job, I think, of kind of emphasizing the flow of information through our brain. So right here we're looking at these different kind of ganglion cells in our retina that are feeding over to the LGN. So this is what we're picking up in the LGN. We're looking at different layers of information. In our LGN, we are going to be looking at really two specific kinds of layers: the magnocellular layers and the parvocellular layers. Magnocellular, as the name suggests, is for big things. It is for big things in your visual field. The parvocellular is small things. And I don't know why I always think about this. I always think of like, so you may have heard of the parvovirus, which is for dogs. I always think P for poodle. So parvo is poodle in my head, and that's the mnemonic that helps me think about what this does, because parvo, poodle, small, this is going to be handling the smaller elements of our visual field. So magnocellular, like magnum, is gigantic, it's magnificent, and then parvocellular is, really small things in our visual field. So our LGN, this part of our brain, is going to take some of that information. What are the big things that we've seen? What are the little things that we've seen? And it's going to distribute those things to our primary visual cortex. And so here you can kind of see that the magnocellular layers of our LGN are going to move to one spot of our primary visual cortex, and the parvocellular layers are going to filter into a slightly different part of our visual field as well. We have these other areas right here, the parvocellular and the laminar regions. Don't worry too much about that. We still don't know a ton about the intralaminar regions. They're just the cellular structures that divide the magnocellular layers and the parvocellular layers. We'll probably learn more about those in the coming years as we continue to study this stuff. So basically we have these two layers. Now the magnocellular is going to be used a lot in motion detection. Why is that? Because if you think about the things that we detect that are moving are almost always big things, because they take up a big part of our visual field. When we are trying to detect what the structure of something is, like let's say to try to detect, what a specific word is. We're going to be paying attention to the form, which is usually a very small, detailed, centric type of thing, which is handled by the parvocellular. All right, so that's all that stuff. We're gonna flesh some of these ideas out as we continue in this video. So the LGN I've been talking about is this structure right here. So after we go through the optic chiasm, now we're going to the lateral geniculate nucleus, which, yes, that is a mouthful. But this, whenever I talk about it in my classes, I usually like to phrase it as it's kind of like a bus stop, where we have all this information coming from our eye, coming from this junction over to this bus stop, and then it's going to get on the bus and it's going to go to separate areas of the brain. The magnocellular information is going to come in, and it's going to go to a certain part of that bus stop, which is going to go to a certain part of the brain, handled differently than the parvocellular ticket holders, so to speak. So this is part of the thalamus. The thalamus is a structure that we're going to see a lot this semester. The thalamus is basically a sensory gateway. So when we talk about the thalamus is going to be part of our brain that helps us decide what information do we want to process and what information do we want to ignore. So if you're thinking about, just for example, you're hanging out, you're trying to go to sleep, but you can't stop focusing on something that you're hearing. That might be because of your thalamus. Your thalamus is letting in all that auditory information. Some people are really good at blocking out those things, which basically means that their thalamus is really good at regulating what information comes in and what information goes out. So this is all just to basically say that the LGN is part of this sensory gateway. And the sensory gateway isn't just specific to vision. We're going to come back to it when we talk about hearing. We're going to come back to it when we talk about motion as well. Or sorry, not motion, but movement and touch and feeling things tactily. So the LGN, the lateral geniculate nucleus, is going to be the spot in our brain where the optic nerve is finally going to come and feed in information. So we have from our optic nerve, it's going to pass over some of those things, but those axons are going to latch on to different areas right here. And so here you can kind of see that we have these 6 different layers. That's because we have 6 different layers. 3 of them are going to be magnocellular, 3 of them are going to be parvocellular. And then in between them, we have these interlaminar regions. But don't worry about the interlaminar regions yet. From there, we have what's called ipsilateral and contralateral, and these are words that I mentioned a little bit earlier in the video, but ipsilateral just means that it's on the same side of the body that is handled in the brain. Contralateral means it's the opposite. So for things like movement and touch, that's always contralateral. But for vision, we see that some of the stuff is ipsilateral. So, for example, we're looking at, in this case, the right visual field here, right? So we're looking at things over here, and this information is going to be handled by the, if it's the right visual field, it's going to be moving over to the left hemisphere, right? So this spot would be what is called contralateral, but stuff over here is going to be ipsilateral. All right. So, talking a little bit about the different kinds of layers. We already talked about the magnocellular layers. We already talked about the parvocellular layers. I think I misspoke earlier whenever I said that there were 3 different layers. It's just because this is not actually referring to those magnocellular layers. Instead, the magnocellular layers are going to be the bottom 2 layers. They're the thickest, the big cells in the LGN, and the parvocellular is going to be the top, smaller 4 layers. And like I mentioned, magnocellular is going to be responsible for detecting big changes in our visual field. Parvocellular is going to be the smaller layers, and the coniocellulars, these are the interlaminar regions that I was just talking about. We're not entirely sure what those things do yet. All right, so that's all that I want to talk about for this specific video because we're running a little bit long on time. So I'm going to pick up in the next video by talking about the primary visual cortex, also called the striate cortex, and we'll also talk about split brain research. So why is that we know that certain areas of the brain are responsible for handling certain different kinds of information? I will see you there. Bye.